I want to welcome you to the Informatics and Data Science lecture series that NLM sponsors. We're happy to see you in the room, and also those of you we can't see on the video cast, and want to let you know that this will be available on the NIH video cast in perpetuity. There's a sign language interpretation available, and if you're in need of that, please let our interpreter know so she'll make sure that she stays here. I'm happy to introduce Joshua Swamidas, who is an assistant professor of laboratory and genomic medicine at Washington University. He received his PhD in computer science and an MD degree at University of California, Irvine, and he's really focused his research on problems in medicinal chemistry, as we'll hear more about today. At Washington University, his group develops and applies new computational methods at the intersection of biology, medicine, and chemistry. Most recently, his focus has been modeling metabolism. In 2016, he received his first R01 as an early stage investigator from NLM, and I mention that for those of you who are earlier in your careers. This is the next step, right? Early stage investigator. In this particular work, he's studying mathematical models that predict when molecules form reactive metabolites, with the goal of making medicine safer. He's published more than 40 papers, probably more than that by now, with a weighted relative citation ratio of 62. And those publications include a series of papers that use economic theory to better design high-throughput small molecule screens. And that work was extended in collaboration with GlaxoSmithKline and Pfizer. And I'm also pleased to say that he is a member of the National Library of Medicine Study Section, the Biomedical Informatics Library and Data Sciences Review Committee. Rather than read the title to you, I think I'll let him read it to you. How's that? Welcome.
Thank you so much, Valerie, for that introduction. And I'm really pleased to be here. It's an honor to be at the NIH talking about my research. The title is Translating from Clinic to Chemistry with Deep Learning. And you could also have the subtitle there of Modeling the Metabolism and Subsequent Reactivity of Drugs. I'm also pleased to report that all the final work is done. I was also promoted to associate professor, so now we can start saying that. Which is, once again, a real honor at WashU to reach that stage. Today, I'm going to be talking about the first of two grants I was fortunate to get from the NLM, where we were building tools and ways of modeling how drugs become toxic. I'll be talking about that in a moment. But our group really is looking at a large range of things, not just how they're toxic in this one pathway, but also how to understand how that changes in different patient populations. So specifically, the second grant we got was looking at how that changes for children. But then also we do work collaboratively with several people. One of my collaborators, Sid, is here actually. We did some work in virology. There's also a real nice piece of work that Ranjan Kumar, who's about to graduate from an PhD, did with our group that got into Nature Genetics, looking at using deep learning to understand cancer drivers. And more recently, we've been doing more and more work in medical imaging. And I'm not going to talk about that really at all during this talk, but the main reason why I bring that up is that we're using the same methodology across all these different domain areas. It's this new technique which people are hearing about more, which is called deep learning. And it's being used almost like it's a buzzword, but I'll say that we've been doing this really since I started my PhD in 2002 in an NLM-funded group at University of California, Irvine, Pierre Baldi's group. So this is really where I started my training, what we've been doing our work in over the last 15 years. And we were doing deep learning essentially before it was called deep learning. Let me take a moment and focus in on the specific question that we're going to try and look at today from several different angles. So we have here is two different drugs. Both of them have the same alert structure. It's this little 5-membered ring with a sulfur on it, here and here. And the thing that's a little bit strange about these molecules is that there's like a shading behind it. That's really just the output of our our model on the molecules on an atom level. And you can see here that the sulfur, the S, is circled, and it has a higher value on it on this molecule than it does on this one. The reason why is that this molecule gets oxidized or metabolized, or the structure is changed at that thiophene, at that sulfur, and oxygen is added. This one, that doesn't happen. This is important because once the sulfur is oxidized, that makes it reactive, and it can actually bind to a protein, and it forms then a hapten complex, which can then stimulate the immune system and cause hypersensitivity-driven toxicity. Some of the hardest types of toxicity to predict, both on a patient level and also on a molecule level, figuring out which molecules can become toxic or not. So this molecule has an alert structure, and it becomes toxic by that mechanism. This one does not. And you can see that we can correctly predict with our model, first of all, that this one is metabolized, this one is not, and then also this one, once it's metabolized, becomes toxic. So that's the big goal we're going after, not to do that just for the specific case, but to take the combined knowledge of thousands of studies done over the last several decades to build mathematical models to quantitatively understand the nuances of chemical structure and the nuances of reactivity, so we can take a new molecule and have some educated understandings and hypotheses about what may be happening with that molecule. So let me drill down to one specific example of a model we built. I'm going to go into more detail on this model than we do in most, but this is the basic pattern we're going to be using over and over again, and being able to build several different artificial minds, you can put it that way, or mathematical models that are going to quantitatively summarize large amounts of published data to start making inferences about new molecules. So in this case, we're going to look at quinone species. Quinones are an important subclass of reactive metabolites because the vast majority of reactive metabolites end up becoming reactive by this mechanism. So and the reason why is because there's so many starting points by which that you can actually get to this point of getting a quinone. The basic idea here is you start with an inert structure that gets metabolized in 1 or 2 or maybe even 3 or 4 steps into something that looks like this. You can see that the aromatic part of this molecule has collapsed into alternating single and double bonds. It's what's called a Michael acceptor, which makes parts of that molecule very reactive to proteins and DNA and other sorts of components of cells, and they tend to link. Now, it's conditional. It doesn't happen in all cases, but that is a common first step in making a molecule toxic. Now, in this case here, I kind of, once again, have the contrast between true drugs, one which is bioactivated by this pathway and one that is not. And what we want is a model that can distinguish those two cases. Quinone formation pathways are very complex because it can happen either through essentially one step like this or multiple steps, and this is just an example of a 2-step pathway. And what we want is a model that can really capture all of that and tell us by looking at the starting molecule whether or not it's likely to form a quinone or not, and where in that molecule. All right, so if you go right here, this is the structure of the model we're using. This is just one way to represent it, where we have here the molecule structure, and these are different vectors that we have to describe the molecule at different levels, both at an atom level, at a molecule level, at pairs of atom level, and then all of this feeds forward to predictions at all of those different levels. So you'll notice that there's a hierarchical component to the data. So a single molecule will have several negative atoms that are part of it, and also several negative pairs that are part of it, even though it itself is a positive example as a whole. So we actually collect the data at all those levels. So we'll have a whole bunch of examples that are similar to one another, where we have no quinonation on this molecule, but then quinonation on this one. In this case, we're looking now, we've taken the atom level labels, and now we're looking at that at a pair level, and we're also taking it now and looking at it at a molecule level, and the colors here correspond to this diagram. And one of the real strengths of deep learning is that it can actually deal with structured data like this directly. So we're not collapsing this down into a single Excel file or table. We're going to actually be learning directly off the structure of a molecule. I'm not going to go into a full detail of all the different types of architectures you can use, but most of the studies we're using are variations of a graph convolution. So what we do is that we make a prediction at every single atom or bond, looking at the labels we have and everything that's adjacent to that atom or bond in the actual molecule graph. These are just some of the formulas that are behind that. This is just one way to do it. There's many different ways. You'd have to really get into the method sections of the papers that we've been publishing to really get a full taste of that. But I wanted you to see what it looks like. So how well does this work? Works pretty well. So this is on the quinone data, and here in the gray we have descriptors from quantum mechanics simulations on these. It's a semi-empirical method where you can get things like the electrophilicity or nucleophilicity of different atoms and such. And the reason why we compare to that is that this is an example of a model that we built where there's no other prior model to compare to at all. This is really the first-in-class model. This is the first model you can find in the literature that predicts quinonation on a large range of molecules. But we still want to be able to compare it to something. So one of the things that we did is look at actual quantum mechanic descriptors. And you can see that there's a very large jump in performance from what you get out of quantum mechanics simulations to what you get out of the machine learning model. And also you can see there's a jump in doing this on purely an atom level to doing this on an atom pair level. Still, we're kind of left with displaying predictions on an atom level because there's no clear way how to do that with pairs. And here's some examples of molecules here. The circles are what the experimentally or reported results are for where these molecules are quinonated. And you can see that it's picking out quite reliably as a top pick exactly where on that molecule it's most likely to be quinonated. And if you're an organic chemist and I showed you this without having the predictions or the circles there, it would be very hard to pick out exactly where, because there's actually multiple places on all these molecules where quinonation could happen, but the model is actually able to selectively hone in on, get the exact place where it's actually happening. All right, so another piece of this that we've looked at really carefully is whether or not this can direct how to modify drugs to make them less toxic. So for example, this is a drug that we know is quinonated, and that's a big reason why it becomes hepatotoxic. And we can see two analogs of it from the literature and what the predictions are, and you can see that the ability to bioactivate is extinguished just as we see in the experiments. You can see, here's another example too, where you have the exact same alert structure in two different places. This case, you get a very high prediction. It almost looks like we didn't put a prediction on these negative molecules. That's just because it's so good at telling us that it's really not going to be quinonated. And you can see that it really can correctly identify the fact that that's not actually a quinonated molecule. And you'll notice here I keep on pointing to this idea of structural alerts, or these structures that are already known ahead of time to be bioactivated. One question we looked at is whether or not within a particular structural alert, controlling for structural alert, can we actually pick out which molecules are being bioactivated or not? That's important because we want to really see that the molecule is really picking up on subtle nuances of structure, not just using the hammer of using stuff that we already know to figure things out. And so while chemists would be able to say thiophene are dangerous, we don't want to normally put those in molecules unless we absolutely have to, that neglects the fact that actually the vast majority of thiophenes aren't bioactivated. Can we distinguish which ones are from which ones aren't? And the same is true for quinones, because in general, for quinones, you can't avoid all structural alerts. And eventually, every drug is going to have a quinone alert in them. So how do you distinguish when? What's the context by which that alert becomes a problem? So, for example, we looked at phenols. So phenols have this basic structure here, and this is an AUC curve showing how well we can separate the quinones that are bioactivated versus the ones that aren't. You can see we get really high accuracy. It's 95% or so AUC, and here's some examples of some positive cases. And then here you have circled in red the actual alerts in these cases, and you can see that it's clearly picking out the cases where it's not actually being bioactivated. Here's another structural alert. You can see that it's picking out, once again, really clearly the cases where it is bioactivated, not just telling you that the molecule is bioactivated, but clearly showing you where the molecule is bioactivated. And in this case, it's showing you where it's not bioactivated. Another question we can look at is the molecules that don't have structural alerts. So these are the ones that aren't obvious. These are the ones that when you look at them, they don't match a standard pattern that you would have right from the get-go saying, that's a concerning place to structure to put in a small molecule. And we want to find out, does the molecule actually correctly identify in those cases too, things that would have just been missed by standard filters. And I'll point out that there's quite a large number of molecules that fit this pattern. So we're able to find over 400 examples in the literature where a quinone forms from something that is not a structural alert. And this happens quite a large number of times. We see around 70,000 cases where there's a potential for this to happen, but we don't really have a good way of deciding when it's happening or not. And you can see here we get very high. And you see, once again, it's really clinging to the side of the plot here, which tells you it has a really good recall. And here are some of these examples too. These are not well-identified structural alerts, and yet we can pick out exactly which ones are forming quinone and not. Now we can extend this to other types of pathways. So this is another type of modification that can happen in someone's liver, where a molecule goes to actually forming an epoxide. Epoxide is a three-membered ring. I'll show you a couple examples of pathways of that right here. So this is the canonical path, where you start with something that looks a little bit like an aromatic or a double bond, and you get a three-membered cyclic structure that has an oxygen that becomes reactive. And sometimes in the data, we'll see that it gets hydrolyzed like that as two adjacent hydroxyls, and sometimes we'll see it where it gets hydrolyzed, but it also gets conjugated to GSH or to a protein. So we were able to build a database of 780 molecules and ask the question, 780 positive molecules with matched pairs, I should say, and ask the question, can we actually use this to understand structural alerts better? So this is another example of a structural alert that becomes toxic. In this case, we know that if you get epoxidized here on these double bonds, it's very likely it's going to become toxic. It will usually get conjugated to proteins. This is a set of, I think, about 9 drugs. I have to count it carefully to be entirely sure, but all of them have furans in them. If they get epoxidized, they're probably going to be associated with hypersensitivity. If they don't, even if they are associated with hypersensitivity, it won't be by that mechanism. And we can ask which of these are actually going to be epoxidized or not, overlay the predictions on it, and we find out that it's able to pick out the 2 known drugs out of this large group that are actually bioactivated. We can go back again to thiophene, which is an original example. This is another case where you're X-oxidized, or if you're epoxidized, the chances that you're going to become conjugated are very high. And we can see that we can get a very good separation again between the ones that are bioactivated and not bioactivated. So this has actually led to a tool that's being used by one of our industry partners that they're actually using internally to help understand why the drugs are seeing become toxic or showing signs of conjugation actually are becoming that way, and how to modify the drugs to make them less toxic. Now, I've been talking primarily about this issue of how drugs are metabolized or bioactivated, but we should also pause for a moment and consider the second piece of that, because reactivity is similar to metabolism in the sense that it's conditional on complex factors within a molecule. Molecules are not always going to be reactive if they're quinonated, for example. They're not always going to be reactive if they're epoxidized. So what we want to do is be able to have a model that can figure out how molecules are going to be metabolized, produce the metabolic structure of the metabolite, and then predict if that metabolite is going to be reactive, and use that and chain all of those models together to then see if that bioactivation is likely to be something that's a problem. So this is another first-in-class model that we were able to build that looks at predicting whether or not metabolites are likely to be conjugating with things in our bodies. Now, what's distinctive about this is that we didn't look at only one problem at a time. We looked at several problems at the same time. We built 4 separate data sets, and we were able to build a model that learned across all of them at the same time. So you can see here there's DNA and there's protein, which makes sense. You don't want things that conjugate to DNA because that can lead to mutagenesis and have a whole bunch of bad effects. You also don't want to have things that bind to protein, because that can also likewise creates lots of problems, both by inhibiting the actions of those proteins, but also through hypersensitivity-driven toxicity. GSH is an important case too, because that's one of our body's natural defense mechanisms against toxic metabolites. It's also how we detect them in vitro. So it's common to take a molecule, incubate them with microsomes along with GSH, and see if we can find conjugates of GSH as a signature of things that are potentially toxic. Cyanide is used in the same way as GSH, is while we don't have much cyanide in our liver cells, at least hopefully we don't, the reason why we use that is that's used as a small molecule proxy for DNA. So it's one of the ways how we detect in vitro whether or not something might be conjugating to DNA. So a couple things about this. We have a lot of data on cyanide reactivity. We have a lot of data on GSH. We don't have that much data on DNA and protein conjugation. And that makes sense because to really establish that, you have to do much more involved mass spectrometry experiments and so on and so forth to really establish how things are conjugating. So there's a much smaller amount of data we have in those cases, even though those are the cases we actually care more about. We did a controlled study where you take the protein data alone and try and model reactivity, and it doesn't work very well. So one of the real strengths of deep learning, however, is that it can, in an automated way, take very closely related problems, like these are very closely related, to learn how to map between them, how to transfer information about one to the other. So while proteins are not the same thing as glutathione, for example, there is an analog there. We don't know precisely what that is. We can't tell you up front exactly what molecules are going to bind to proteins and not GSH, so you don't want to treat them like they're the same thing. But we do know something binds to GSH. It's also very likely to bind to protein, and we want a model that can basically automatically learn how that information transfers from one to the other. So that's why there's strength in actually looking at all these problems together. So this is our results. Once again, we didn't have a comparison model because this is the first time in the literature this has really been done. So we compared again to what we can get off a quantum mechanics model. And there's some really good theoretical justification for this because for the most part, these molecules are acting as either soft or hard nucleophiles or electrophiles. And we can actually measure that in a quantum mechanics simulation. So we did that, and that's what all the blue bars are, and the green bars are the output of our model. You can see our model reliably outperforms what you get by looking at a pure quantum mechanics approach. Notably, too, is that we're getting pretty good performance on the protein. We're getting pretty good performance on the DNA, too, equal with GSH, which, like I said, is really surprising. I don't have the data to show you here. If you did it purely on just the protein, you would not see results like that. You can see also, too, it's not just copying between all of the molecules and just putting the same result for all of these. You can see cases where you get the same prediction on everything. So here you can see a low prediction for both protein and GSH. Here you can see a higher prediction for both protein and GSH. But you can also get examples where it flips. So this is an example where you get a low prediction for protein and a high prediction for GSH. That's important because this might be an example of a molecule that would be falsely labeled in an experiment as potentially toxic, but on the other hand, if you look at it in terms of its ability or propensity to bind protein, it's not as likely to bind protein as you might initially think. And it's really efficiently binding to GSH, which is how our body protects against these things. So maybe this molecule isn't nearly as toxic as we'd think just by looking at a simple in vitro screen for GSH binding. We can see the opposite of that too, where you have here molecules that are predicted to be high binders to protein and DNA, but low propensity to bind with GSH or cyanide. So these are cases where molecules might look entirely okay in an in vitro screen, but there's good reason to think that maybe we need to do a more expensive or difficult screen, either a radioactive binding experiment or actually doing mass spec to really figure out if it's actually binding protein or not, because in these cases, it's possible they could be false negatives in the quick in vitro screen. So one of the other interesting things we came up from this, which was surprising and important, is that we trained on qualitative data, if you remember, whether or not something binds to GSH. But there's also a lot of intrinsically good negative data wrapped up in this as well, which is a good thing. One of the things that we find out from this is that if you take the model's output, which has been trained on quantitative data, and use it to predict the reactivity of data it has not been trained on, data that is quantitative that's been done experimentally, that we get extremely high correlations. We're getting an R squared across an entire data set here of 0.91, which is a really exciting result. It means that even though we're training on qualitative data, that in some domains, in domains that are actually relevant to medicinal chemistry, this is actually a quantitative prediction. So that's encouraging. So you can see this is true both for subclasses. So up here we have aldehydes, right here we have benzoquinones, and right here we have esters. And you can see all of them together. We see a really strong correlation. And once again, this is data that the model did not see during training at all. and we're getting these really good results. And this is also extrapolating to a much broader range of chemistry than typical Q-star models would. All right, so what we've done in our group is we've built a large range of models to cover several different pathways like this. So we've done epoxidation and dealkylation, oxidation, reduction, UGT conjugation, all these different types of reactivity. We're about to submit a paper where we cover around 90% of phase 1 reactions. And I presented at the last Gordon conference in metabolism on how we've been able to extend this also to a very large range of phase 2 conjugation reactions too. And we have in preparation right now a manuscript that's starting to string them together. But what I wanted to do is actually talk about our first paper where we've been starting to actually string these together to look at both the formation of a metabolite and predicting its reactivity to be able to predict whether or not a molecule becomes hypersensitive, toxic by hypersensitive bioactivation. So this is a paper we just had come out last month in a chemistry journal. And what we're going to be looking at here is nitrogen dealkylation. I'll show you some examples of that in a moment. But one thing that's distinctive about this paper is the first time we're able to start with a molecule, predict how it's metabolized, predict the structure of the metabolite, and then look at the reactivity of the metabolite and see if that correlates with what we see with toxicity. So here's some examples of what I'm talking about. So first of all, you can see here there's a nitrogen and there's a carbon chain attached to it. That's what we mean by dealkylation, because what happens is as that bond gets cut, this side gets turned into an aldehyde. There's a double oxygen bond that happens there. And this part goes away, and usually it's not toxic. But what usually is the problem is what happens here, is because that structure can sometimes become toxic. So in this case, what we did is we actually started from the predictions, which you can see overlaid on top of the molecule, and then inferred what all the metabolites were going to be. And we have a metabolite prediction code as well in our group. And then we multiply that times the chance that that aldehyde is going to be reactive. And we just multiply that times our reactivity prediction. And then we ranked all the metabolites. And this is just the top list of hits. And you can see here that the model says there's about a 75% chance that this is going to be a reactive metabolite off of this molecule. And corresponding, in contrast with the next molecule on the list, which is just 0.1%. So you can see here that it can distinguish between both the known metabolites and the non-observed metabolites, but more importantly, can identify the metabolite that's most likely to be reactive off this molecule, which once again corresponds with what we actually know for this case on the experiments. It does the same here on this molecule. Now, we applied this in this case too, and we got a really interesting result that one of the strengths of or one of the reasons we've been excited, I would say, about the grant that we were able to get funded through the NLM is that it gave us the ability to work with an experimental collaborator to start validating interesting things that came up. This is one example of them, which we've been able to get experimental validation on, and we're about to submit the paper on this as well. So terbinafine is an interesting molecule because when we put the molecule in, as you'll notice, it doesn't actually predict the formation of this reactive metabolite. What it instead says is that it's going to be a two-step pathway. Instead of going from here to here, it's going to go down this pathway. Terbinafine is interesting too because it was on the market first as a topical and then as an oral drug for a long time before people, about 10 years before people were able to figure out why it became toxic. Part of the reason why is that that two-step pathways of forming reactive metabolites are often much harder to detect in the experimental approaches. Usually they're tuned to be able to get the first generation of metabolites to have a harder time with second generation ones. And so if you do the experiment in a standard way, it turns out that you won't pick this one out. Another problem too is that reactive aldehydes don't tend to conjugate terribly well with GSH. So there's several reasons why the experiments ended up missing this. And we're able to see that maybe this is part of the reason why. And that's a mechanism we can propose, and it turns out that's exactly what we see when we look at the experiments. That it turns out to be a two-step activation instead of a one-step, and it also doesn't bind efficiently to GSH. It binds much more efficiently to protein and to other reactive metabolite traps, which my collaborator Grover Miller in Arkansas was able to demonstrate. So that's an exciting result. It shows how computation in collaboration with experimental approaches can really work to get these really high value with clinical significance gaps in the current approach to screening for these molecules. There's no way to really get this molecule off a purely experimental screen. You have to know what to look for. And the way to know what to look for instead of just blindly testing for everything is having a computational model to tell you what's most likely. This gets to how we've been thinking about translational medicine in our group. In a lot of ways, this has been a very low-level study of chemistry, which seems even below what people think about in biology. We've been talking about quantum mechanics, which, that's even below what we think about in chemistry typically. And that's, I think, what's actually really interesting about this problem, because we can actually go and trace features of the quantum chemistry of a molecule, trace that up to how it affects chemistry, how it affects biology, and ultimately how it affects things in the clinic. This is multi-scale modeling at its most interesting, I think. And the typical way I think we think about translational science is usually trying to just get things over the fence towards basic science, translating from basic science to bedside. There's been a little more intention about going from insights we see in the clinic to understanding what the mechanisms are. I think that is an important, valid way to think about it, but in our group we've been thinking about it instead of as a quest to just get something over the fence, think about it from a computational point of view of how we can have a lively and rapid conversation back and forth between the two domains, which is something that we have the ability to do as we get better and better clinical data and better basic science data. So how's that playing out in this case? So first of all, aldehydes have been recently proposed as an important toxic metabolite. What I just showed you is actually controversial in the field because the way how we experimentally look for reactive metabolites aren't good at picking up aldehydes. And so it's been recently proposed that they're an important toxic metabolites that's missed. Now on the clinical side, there's several drugs that are hepatotoxic by hypersensitivity-driven ways, but we don't know why. We don't actually know ahead of time what the bioactivation mechanism is. So we can take these two things arising in two separate discourses and ask now, is there a connection between these? And can we ask, are there drugs that are toxic by this mechanism that we can identify computationally? The first step of that is what I already showed you with terbinafine. Now we can actually look to see whether or not we can see other drugs that fit this pattern that we can study deeper. And so we did that. We looked at molecules that are known to produce aldehydes and looked to see if their aldehyde was predicted to be reactive. We can identify actually quite a large number of drugs. We're talking maybe about 100 or so. And we can then filter this to find out the ones that are hepatotoxic and then filter out the ones that are hepatotoxic by mechanisms that we already know about. And from there, we can get several really interesting cases where this is a potential hypothesis that could explain why that drug is toxic. We can take this further now, too. One of the reasons why it's been hard to even establish whether or not a drug is toxic in a clinical context is you can take a drug through 1,000 patients and never see the toxicity arise. It's idiosyncratic often. Well, idiosyncratic is just a fancy way of scientifically saying we have no idea why. There's no pattern behind it. And the question is there a pattern behind it that we haven't seen yet? And of course, then, almost by definition, it means that it's no longer idiosyncratic. But so then in the end, maybe what we're trying to do here is shrink what the idiosyncratic nature is. And you might say, why does it matter if it's only affecting 1 out of 1,000 patients? Well, 1 out of 1,000 patients is enough to get a black box warning. It's enough to get a drug pulled from the market. It's enough to deny the 999 other patients that benefit from that drug access to that patient. It turns out that that's how severe of an issue it is. It turns out also, too, that maybe it's 1 out of 1,000 among adults, but it's a lot higher among children. And if we start giving it to neonates or to 2-year-olds or 3-year-olds to treat something, maybe it has a much higher burden on them. And if we don't understand the mechanism, we have no idea how to even really test for and look for that. So yeah, we really do want to understand it, even if it's 1 out of 1,000 cases, even if it's 1 out of 10,000 cases, we really want to understand it. Because if you're that 1 out of 10,000 case, and it puts you in a situation when you have severe reactions to it, it matters a lot. It doesn't really matter to you how many other people are in that situation. So we do care about that. And as we understand the mechanism, it can give us hypotheses about why it is that some patients are affected and others aren't. Now, one of the things that comes out of understanding this mechanism is that we have a way to detoxify aldehydes in our body in addition to conjugating with them. We have an enzyme, actually a set of enzymes, I'm just going to simplify it a little bit here, called aldehyde oxidases that are really efficient at finding aldehydes and turning them into carboxylic acids. They add another oxygen, makes them safe, they're not reactive. And that's generally why they've been ignored in drug development for such a long time, because you just assume if you see an aldehyde, well, the aldehyde oxidase will take care of it. But we also find out is that there's a large number of drugs that are on the market, commonly prescribed to patients, that inhibit that pathway. So one possibility is that part of the pattern that would make these drugs idiosyncratic is that it's not just that you have to be metabolizing and producing that aldehyde, you also have to be inhibiting your detoxification pathway somehow. So we can now use humans as a model system in the EHR to look for cases where there's patients that are prescribed that drug that forms a reactive aldehyde alongside drugs that inhibit the detoxification pathway and see if they have a higher incidence of drug toxicity in those cases. So that's something that we're in the middle of doing in our group right now. And so now we're getting to the point of going all the way from quantum mechanics now to the clinic, which I'm saying is really the exciting thing about this work, just on a conceptual level, and then also how we're able to engage this on a month-to-month basis in our group. Moreover, we can start thinking about important cases. So for example, there was some HIV drugs in the prior slide where we can think about how we can modify the drug to make it less toxic by extinguishing that activity. It can happen either at the level of making it more difficult for that molecule to be metabolized, or less likely to be reactive even if it is metabolized. I really do think in many things in basic science, and especially in our work, that clinical data really is the future. It's becoming available at a larger scale. So in our group, we're looking much more at EHR data. When we start thinking about resubmitting this data, these grants, it's going to be including large efforts to use humans as a model system, looking at inpatient data to see how when we perturb in these natural experiments, not actually having physicians change what they're going to do based on our models, but just seeing what they're going to be doing anyways as natural experiments to see if that gives us insight into the underlying mechanisms of bioactivation. And we can also look at VA data. This data is becoming available once again at a very large scale. Just at WashU alone, we have about 500,000 hospitalizations that we can look at over a 5-year period. Just a 5-year period in one hospital, 500,000 natural experiments. Isn't that amazing? And so there's a scale of data issue there, there's a complexity of data issue here, and I think that is actually where a lot of major advances are going to happen. And it's going to bypass entirely the problem of figuring out how to interpolate from an in vitro study to in vivo. It's going to bypass entirely the problem of dealing with, going from a model organism system like rats or mice to humans, because we're going to be dealing with humans directly. Once again, to be clear, not experimenting on humans, but seeing it as a natural experiment, because there's already manipulations happening just in the natural course of their care to understand what's going on there. Of course, you can think about now how there's ways how that can start informing clinical decision support too. Turns around, you can tie about 20% of Medicare costs to liver injury from drugs. That's a massive amount of money. That's a minimum around $200 billion a year tied to this problem. And if there's better ways to manage it, that's going to be important. That's going to be valuable. There's also an opportunity to look at this in the context of clinical studies. One of the things we can get out of clinical studies is a lot more information about individual patients. It's common in clinical studies, for example, to get the metabolites in vivo for every single patient in the study. And if we can reconstruct the pathways and see how alterations in the metabolites on a patient-per-patient basis and infer the internal pieces of those puzzles, there's ways to actually understand and get biomarkers for toxicity for patients early on in a study, which can give us now rationale for understanding why it is that certain patients in a study are toxic, which ones are not. And once again, also using that then in clinical study design. That's a major challenge facing drug development at this point, is how to actually design these studies so they're robust to idiosyncratic effects like this. And this is one strategy to do that. The other work that we're doing, I have a K award out right for one of my, not a K award, an F award, I'm sorry, for one of my MD PhD students who's working on this, looking at the FDA adverse event reports where we can actually look for differences in reporting, the relative odds ratio in reporting for different ages for hypersensitivity-driven reactions. We can look to see where there are sensitive periods in development. For example, is there a drug that seems to just be more toxic among 5-year-olds than it is around teenagers, or more toxic among neonates than teenagers, and start understanding based on how the enzymes are shifting why that actually is, really getting down to mechanism in a way that you could never ethically do a clinical study. We're going to have to be doing something that takes these clinical observations we're getting, building up actual data-driven hypotheses that we can then validate in the lab and have now this coherence of being able to explain and understand why things are different for some patients versus others. So maybe it's possible. Well, there's two ways to think conceptually about what we're doing. On one hand, you could say we're trying to reduce the number of idiosyncratic reactions just because even if they're rare, we're going to know why, so they're no longer idiosyncratic. The other way to think about it is that we're trying to understand them. So maybe as a class, idiosyncratic could take on a new meaning. It's the ones that are just rare, but we are starting to actually make them tractable to understand on a mechanistic basis why they actually are toxic. I want to also point out, too, that this emphasis on mechanism is something that might seem like a non sequitur when we start the machine learning, and in deep learning in particular. Often deep learning is or functions like a black box where you just throw lots of data at it and you try and get a prediction out of it at the other end. That, I think, is a mistake. It's almost an anti-scientific way to use machine learning. In the end, science in Latin originally meant understanding. What we really need is understanding. And when we think about that, on some level, we want to understand mechanisms. And I think what's exciting about machine learning, when it's used in a scientific way, is it can do exactly that. It can give us understanding, and it can open our ability to actually see mechanisms because we can see patterns in the data we did not already see, and there's ways to then directly take that into the lab and test them. So as I'm closing up, I do want to acknowledge several people who have really made this possible. Most of the group and the work in this group has really been done by 3 phenomenal students in my group that are all graduating probably over the course of this next year. Tyler is the first PhD student from my group, and then Lena and Matt are both MD PhD students who are just doing phenomenal work. They're going to be some of the best trained machine learning students coming out that also have training in medicine, which is particularly exciting. They're going to be graduating and being on the market in just a couple years. And also a lot of the work we've done, my initial work was at the Broad Institute with Paul Clemens and Josh Baker, and I just want to thank them for hosting me, and then obviously Grover Miller, who's my major collaborator in Arkansas, and then also the NLM for funding this work generously. And I think hopefully we can really do some significant things here to make tractable this area that people for a very long time have thought is intractable. So thank you very much for your time.
We have time for questions, so have at it.
So I think you raised your hand first. Go ahead. Yeah, so I didn't get that into detail. The question was, there was a desire to know a little bit more about the specific architecture he's using and how that becomes explainable and understandable to figure out exactly what's causing things. So I'm just repeating it so people online can hear the question. That's an excellent question. I'll point out that there's a lot of resistance to making deep learning models interpretable. I would say that's not something intrinsic to the math. It's not something intrinsic to the method. It's just more of a cultural difference. It's the difference between scientists and engineers. Because it's entirely possible to make interpretable deep learning. It's actually fairly easy to do. It's just not how engineers are trained to think about deep learning. So it's a good thing to resist any notion of a black box method. So we've been thinking about this a lot, and I can't actually get into all the details of this, but what I'll say is one of the things that's been really important to us is instead of making predictions on a molecule level, we've been wanting to make them on an atom or a bond level. So the way how that becomes interpretable is that that's the language by which chemists communicate. So you can see here, we do make a prediction at a molecule level, but we also make a prediction at an atom level. So we can now paint the molecule, and the model is telling us when we do this that this is the part of the molecule that's causing it to have a high prediction. And that's the language, actually, by which chemists actually talk about these molecules. So it ends up being a way to make the model actually be able to communicate in a way that chemists will really understand, and that is actually interpretable to do it that way. Now, there's a second question about how do we know, well, why is it that that molecule is actually becoming reactive at that point? There's two approaches to dealing with that. One is that you can actually figure out which of the input attributes to a molecule are important. So one of the things that we did on the epoxidation study is exactly that, and we found out that there's a specific quantum mechanical feature that is really critical to making that prediction. It's called pi-p bond occupancy, or basically how much of the electrons are sticking out into the, outside the plane of the ring. It wasn't really known that that was an important predictor before, and it turns out that there's really good quantum mechanic justification for why that would be important. It turns out that there's a reason about how the actual enzyme substrate complex forms, why that should be a good predictor, and we can actually validate that now at a lower level. The other way we can do this too, and this is once again getting back to the language of chemists, one question that they'll have about this structure is how do I modify it to extinguish that? What if I can't get rid of this structure? Is there anything I can do over here, here, or here that I can actually modify that molecule by to actually change it over here? So we can do something called a perturbation study where we perturb the input in ways that make sense to an organic chemist and thereby understand how to change it to get rid of that behavior. So that's another set of studies that we have that have not yet been published yet, but we're working on where we can do that in an automated way. And once again, present it in a way that an organic chemist can understand. I'm like, oh, okay, that's how I modify the molecule to make it less tuned to go down that path. So it's a combination, I would say, of understanding really the domain very well, knowing the domain language of that domain very well, and then also being able to clearly build a structure that allows us to map clearly between what is happening mathematically and how experts there are thinking. So there's a couple other questions. Yeah, you had a question? Go ahead.
You have a question about the input, because it seems that you use pairwise information and atom-based or some other, I don't know. My question is, do you use like
So we've published, so the question is like, what exactly is going as input to the model? And that's a good question. And I can't answer it in well, the general answer is that it really depends on the problem, and there's a very large range of stuff we did. If you look at each of our papers individually, it'll be clearly mapped out exactly what goes into it. Now, sometimes we're using quantum mechanics simulations. So you can, for example, do a quantum mechanics simulation on the molecule, and then use the output of that simulation as input into the network. So not on this particular model, but I'm just saying in general, that is one thing that you could do, but not in this particular model. So another thing you can do, which we did do in this model, is look at the graph structure and use attributes that describe what's going on in that that atom, and then what's going on one atom away, what's going on 2 atoms away, what's going on 3 atoms away. So you basically give it a picture, a mathematical picture of what's going on in concentric circles outside of that spot. That's what's described right here. So right here you have a little node, a convolutional node, that takes in the self, and then everything 1 away, everything 2 away, everything 3 away, and it sums things over things that are those steps away. So it's a convolutional approach. So you can do that as well too. In this case, we're just representing it differently, and that ends up being around like 375 descriptors, given the specific way this is structured in this case. And then that's what the input becomes to the network, and it's taking the structure of the graph into account as it does that. Yeah, so we certainly compared to the fingerprint way. It doesn't work very well. Actually, it works pretty poorly. Part of the reason why is that fingerprints do not actually tune well. So you basically kind of stuck with the strengths and weaknesses of that fingerprint representation. So we've published a lot. I have no opposition to it. During my PhD, actually, I did a lot of work on fingerprint-based methods. But they're really tuned to understand things in a quick way about a molecule, but they don't usually give you mechanistic information without a lot of finagling. This avoids that problem entirely by going straight to it. So some people would even call this a neural fingerprint or a circular fingerprint. So there is a mapping even between this to that type of theory. But if you think about it less as a fingerprint and more as a network, you can find out there's actually a large number of architectures that don't map well to fingerprints that you can apply here. So we just published a paper in January. So we just said last month, except for we're now March, so about 6 weeks ago, where we looked at a large range of architectures and showed actually how we actually introduced a new architecture where we can really efficiently propagate information across a molecule. We can show how all of the more convolutional approaches, even like this one that I just showed you here, really fail on some important problems in chemistry. And how by doing message passing across a molecule, you can actually solve that problem to be able to understand aromaticity and conjugate systems and even quinonation at a much better level. That's just not what I covered today. So we're doing a lot of work also on the actual fine-grained details of the architectures. So thanks a lot for the question. You had a question too, right?
Just to be a bit more precise with the computer side, how big is the computation? What kind of language do you use or libraries? Can you be a bit more precise?
Yeah, so how big is, so the question was, can I be a little more technical on the computational details? So of course, almost everything I showed you here is available as publications. So if you go to my website too, you'll see links to them all. So that's the best way to get the mathematical details. In terms of some more of the information, when we're not doing the clinical data, these are relatively large data sets, but they can usually fit into memory on a single machine. What really makes it computationally difficult is when you start doing cross-validation across large data sets. So in that, we use a cluster for it, but at least everything fits on one machine. That's usually the case, though that's not always the case. So we've also been starting to use these methods to actually create a new level of theory in quantum mechanics, where we train on very large data sets of quantum mechanic data to predict the quantum mechanic outputs. So those actually do require doing incremental training where you can't fit everything in memory. When we, I just may answer one more question just about what the tools are that we're using. When we first started, it was an in-house library that we built because we started doing this back in 2011 or so, or 2010. So things like TensorFlow did not exist. And so we actually built that and we had that in-house and we developed them. That's what pretty much everything in our group had been until about a year ago. And that's because right now with the explosion of deep learning, there's actually some pretty strong libraries. So we use TensorFlow for the rest of it, and for all of it right now. But that's a big shift. Go ahead, you had another question?
The question is less expensive or less heavy than doing the quantum mechanical calculation?
Yeah, it is definitely less expensive than doing the quantum mechanical calculations. So that's one of the reasons why we think there's going to be an exciting opportunity to actually use these methods to replace large-scale quantum mechanics, where you can, if you have a large number of organic molecules, to save 100,000 organic molecules, it is not trivial to get quantum mechanical simulations across all of them. However, applying a neural network to all of them is, or it could be, trivial. So that's what we're working to do right now, too, in our group. Great questions. Yeah. Go ahead. So larger microbial products could be potentially put in. There's going to be some major caveats. So in terms of the method in general, the answer is yes. In terms of these specific models, they're going to be usually trained on things that look a little bit drug-like. And so the issue isn't about the method, it's more about the particular data sets. And in general, if you have a very large microbial molecule, it's not usually going to be metabolized by P450s because it's not going to really be able to fit in there. So that's not usually going to be the fundamental problem you face with it. So that's the good news. So I wouldn't worry as much necessarily about this type of toxicity with a microbial molecule. Now, in terms of Other applications, so for example, if you want to get a quantum mechanical simulation done on a large microbial and small molecule, that's actually very hard to do right now in a reliable way. And we are trying to build neural network models that would be able to handle that extremely well. So that's, for example, what we just got published just about 6 weeks ago, so a couple months ago, showing how the methods here that I just presented here don't actually transfer very well to large molecules. But when you start putting in this message passing ability, it does actually start to work on these molecules, and you can start picking up very large range effects on molecules that you can't get if you're just looking at a small portion of a molecule. And if that's something you actually have an interesting problem that you're working on, please do reach out to us, because we're really eager to form collaborations and work especially with postdocs and students to have the right questions that are there too, where we can take and develop something on this and actually have giving you a tool, even early access before it's published too. Yeah, so what I'll say is that we're very attentive to that. So one thing that happened when we looked at the epoxidation model is we saw that it was really good at doing this for things that were human synthesized, but didn't work so well for things that were natural products, and which often have epoxides in them. And this is also true for the reactivity stuff, where it would kind of just assume, and if a naive data set, it would just say that everything that has an epoxide is reactive. But there's a lot of epoxides that are not reactive in natural products. So what we did is we just augmented the data set. We got a whole bunch of natural product data as true negatives that we could actually validate and put that in. So this is once again where the domain knowledge becomes important. If you just approach this from data into black box out, I don't really care about the domain, you're going to make these types of errors. But we're very attentive to that. And so not even that we have it all figured out. I mean, that's why we're in conversation. That's why we care about collaborators in these domains too, because we want to learn from you as we do these things so we can basically move this forward together. So like I said, I mean, it sounds like you're really thinking about this deeply. Please do reach out to us. We'll have you working with a student in my group and giving you early access to models and to really being able to contribute on that too. Go ahead. Yeah, so once again, this gets to a very recent paper we had that I keep on pointing to. This is an ACS Central Science. It's open access. If you just look it on your phone, you'll find it really quick. It's called, it's called a local model of aromatics and conjugate system size. So that's the name of it. So yeah, we definitely looked at that. It turns out that in a lot of problems, the depth isn't really that necessary. So we're testing that all the time. And this is also where it gets to the whole issue of being an engineer versus a scientist, because as an engineer, you want to make the most complex, cool thing you can go and just go off after it. And the black box works and you're fine with it. In the end, as a scientist, I want a parsimonious model. I want the simplest thing that works solidly. And I want justification to go to a higher level of complexity. And I want to understand what it is about the molecule that makes it require that type of complexity. So then I'm going to understand something about the structure of the problem. So what I would say is the big advance that happened recently is we identified what exactly it is that requires us to go to that higher level of complexity. And it really is going to be non-local features that become important. And in most cases of chemistry, non-local features are not important. Occasionally they are. And so that's, I think, where it starts to become interesting, because now we can actually do models that are local versus ones that are non-local and test to see to what extent things are driven by non-local versus local features of chemistry. And it turns out, as we expect, the vast majority of stuff is local. That's why substructures make sense, or active groups make sense. It's like the basic logic you learn when you're in organic chemistry. But it doesn't always work. And so that's what we're able to start now using as a microscope to really understand the true structure of underlying problems, rather than just using things as a black box. Go ahead, Jane. Yeah, so the question is, how do you deal with cumulative errors, because we're using several models at a time to do that. So what I presented to you was a simple untrained model where we just multiplied two probabilities together, but there's a question if errors are correlated somehow, that's actually not a good estimate, and I completely agree. So what's happening right now, really Tyler, his capstone project that he's working on right now is to use machine learning to solve that very problem, because we actually know the answer at the very end too in many cases. So instead of just training all the steps, we can use the training on the individual steps as input to a higher level model that trains on the data we have at that higher level too. So you can start learning directly from that how to tune the predictions so it deals with these types of correlations and cross correlations. And so it turns out, I don't have data to present. This is like very, hot off the press. We're not even really on the press yet. But hopefully we'll have something to submit in about a month or two where you can actually get very large improvements and be able to identify actually what the actual bioactivation pathway is when you take the individual models and instead of just naively multiplying them together, you actually build a model on top of that really is good at ranking pathways. So thanks a lot for your time. Yeah.